Good afternoon. My name is Victoria Gray-Isle. I'm the division director for the CRCAIH project of SEED, which is the Community Engagement and Innovation Division. And I'm just very excited and happy to be here. And the fact to see Lyle Best on the last slide is a great introduction to a man that I really feel like has been a mentor for a lot of students and people in this region. And so I want to tell you a little bit about him, but I also want to talk about, we're always pushing for time and everything like that. So what we're asking is that we hold the questions for the two presentations at this time, and at the end of their presentations, then we'll ask for questions. Dr. Best grew up in Wahpeton, North Dakota, and graduated from high school in 1966. He earned his undergraduate degree in chemistry from the University of North Dakota and his medical degree from Albert Einstein College in Medicine in New York City in 1974. He worked many years at Belcourt Indian Health Service Hospital, providing family medical services. And in his last 4 years at IHS, he was responsible for the maternal child health consultation throughout the 4-state region of the Aberdeen area. When Dr. Best retired, and I'd say this is his next career, he moved on to be trained for a year in DNA Diagnostic Lab and Genetic Clinic in Winnipeg, Manitoba. He then began his many part-time jobs and much sharing, and teaching genetics at Turtle Mountain Community College, seeing patients at Dunseith Clinic, teaching residents in Minot, working with the Strong Heart Study Research Program in South Dakota, and looking after a few cows and calves at home. Dr. Best and his wife, Susan, live on their son's cattle ranch outside of Watford, North Dakota. And I'd like to introduce him for his presentation, A Case-Control Study of Asthma, Lessons Learned. Thank you, Dr. Best.
Thank you very mush for those very kind words, and I'm happy to have the opportunity to tell you a little bit about the study we began at Cheyenne River, looking at asthma. I think most of you are probably somewhat familiar with asthma. You probably have friends or acquaintances with asthma. It's generally a lifetime condition, but it tends to be intermittent. There are episodes where you have a shortness of breath and problems, and then it tends to relapse or improve, but it's usually generally lifetime. Sometimes young children will have asthma for a short period and it clears up. Generally it can be controlled, but it does have serious consequences, including mortality in some cases. And it's often triggered by many different effects, sometimes emotional changes, air temperature, exercise, allergens in the air, and so on. It is seen more commonly in families, so that gave us the idea that there may be some genetic factors as well. And it's naturally aggravated by air pollution, tobacco smoke, and so on. This is a community-based initiative, our study of asthma. Ray O'Leary here in the audience was really the instigator of this. She's a registered respiratory therapist and RN from Cheyenne River. And she had been working with Dr. James Wallace, who is a Sanford pediatric pulmonologist, and they developed an intensive educational program to try to improve the control of asthma for children. And they were seeing some good results, but this was not a controlled trial. And so when Amy Elliott approached me and she was going to put in her proposal for CRCAIH, she asked if I had any ideas on the shelf, and I said, well, maybe we should think about doing a study of asthma, thinking of what Ray had started. So what we did is we initially intended to recruit 120 children with asthma between the ages of 6 and 17. We weren't quite able to meet that goal. We did recruit 108, and we matched each case with 2 children without asthma who are the same age. And we did a number of tests and measurements, both of socioeconomic factors, demographic factors in the home, air environmental tests in the home. We collected some blood for testing in terms of allergen sensitivity and general inflammation. And we also collected DNA to look for changes in their genetics that might have some impact on their risk of asthma. So within this is what would be called a case-control study, and just looking at the possible causes of asthma. But within this, we had these 108 individuals with asthma, and we nested a randomized control trial looking at this intensive educational program that Ray had developed, thinking that we could perhaps show and prove, demonstrate in a controlled fashion that this was an effective program for improving care. We weren't able to enroll all 108, but 104 of the controlled case children agreed to take part, and so they were randomly assigned to either usual care, which consisted of some written material, which is commonly given in the clinic, and encouraged to continue to see their usual provider, and the intensive care, which involved more intensive education by our staff, and also open access to an asthma educator. The outcome measure we were interested in was the number of emergency room visits related to asthma or pulmonary problems. One of the things we've talked a lot about, developing infrastructure for research in Indian country, and one of the things that was developed as a part of this was a data safety and monitoring board. Whenever you have a controlled trial like this, or an intervention, I should say, you need to have a data safety monitoring board to make sure that if you're starting to see there's a significant difference between one of the treatment arms, that you need to stop the trial at that point and give that effective treatment or non-treatment to the others. And so this was accomplished, and I think there's been very few of these in Indian focused research, but this was done at Eagle Butte. Unfortunately, we were not able to show a statistical difference in an improvement in emergency room visits for our study, possibly because we didn't have quite the power we thought we would have when the study was started. There were fewer emergency room visits even amongst the controls, and so we had less of a comparison to possibly see. And we also recruited just a few fewer cases than what we anticipated initially. We did see a smaller number of emergency room visits for those in the intensive education arm, but it did not reach the level of statistical significance. Some of the findings so far from, and I'll just review a couple of them. We did collect blood samples, and we looked at measures of their immune status. One of those measures is something called the high sensitivity C-reactive protein, or CRP. You see that on the left side there. We did not see a difference between the two, between the cases and the controls without asthma in this particular measure. Some studies have seen that, some have not. We also measured something called the immunoglobulin E, which is a particular antibody that seems to be mostly related to things such as allergic rhinitis, asthma, eczema, and so on. And this level of IgE was higher amongst those with asthma compared to the controls. We also measured sensitivity to 5 different antigens, inhaled antigens. There was a particular mold called Alternaria, which is a common one, cat and dog dander, the small mites that live on all of us. Some people become sensitized to them, and cockroach material. So the blue columns show the prevalence of sensitization to these different antigens amongst the cases with asthma, and the other column, the controls. So you can see in all of these cases, the asthma, those with asthma are more likely to be sensitized than those without. By the way, this is also a measure of what we call atopy, which individuals who have sensitization to these types of antigens are more likely to have eczema, as I mentioned, allergic rhinitis, and so on. And this is a general condition called atopy. And this also relates to at least certain forms of asthma, not all types of asthma, but certain forms, and that'll be apparent in the next slide or two. So those reports in terms of the immune status have been published at this point. We're in the process of a publication being reviewed, looking at the results of our genetic studies. And this is going to be a difficult slide for you to see, but simply to point out that we looked at 10 different changes in the DNA or the genetics of these children to see if we could show that there might be some changes that increase or decrease the risk of asthma. And these 10 small changes, these are only one single letter of the DNA code. There might be an A that's changed to a T in some individuals, or a G that's changed to an A. And this is amongst the 3 billion base letters that you inherit from each of your parents. So it's a tiny, tiny change amongst all of your DNA, but it can have important effects. And these particular 10, we call them SNPs, or single nucleotide polymorphisms. These have been shown in other populations to be related to risk of asthma. And we did see a relatively similar prevalence of these changes amongst the population in Cheyenne River. You see in the two columns on the left-hand side, both cases and controls. This compared with other populations around the world. The EUR, the European population, and the AMR would be the American mixed race population, which would include indigenous people throughout North and South America. In general, the prevalence of these changes is not terribly different than these other two populations. There are some more dramatic differences between, for example, African populations and South Asians and so on. But generally speaking, there were not any particular surprises here. What we found in what we call a multivariate logistic regression analysis, where we look at a number of different factors to see which ones are independently associated with risk of asthma. At the top there you will see the body mass index, or BMI. It does show significant association with asthma, and this has been seen in other populations as well. As children become more obese, they are more likely to develop asthma, and we saw this in our population as well as has been seen around the world. In addition, those children that have at least one of those 5 antigens that we tested, or be defined as atopic individuals, they also increase their risk of asthma by about 4-fold. And having adjusted those factors, the most interesting part for us was we did see a significant association between those top 5 there, genetic changes. These were all in chromosome 17, the long arm, and these showed a decreased risk of asthma for those individuals that had the least likely, or the SNP, the allele with the less prevalence in the population. In addition to those on chromosome 17, there was another SNP on chromosome 5 that also showed significant association with asthma in this population. Again, this is not terribly dramatic findings in that all 10 of these had been shown to be associated with asthma in other populations, but this gives us some important information. First of all, it shows that Indian people in this particular community are not particularly different in this genetic propensity than other people around the world, and it also helps scientists to know that these particular changes in these particular chromosomes may be good leads to places where medications could be developed and so on, and I'll point out one of those possibilities. So the change we saw on the number 5 chromosome was between 2 genes, and I'm going to skip over the one and go to the second one, the TSLP. This is from thymic stromal lymphopoietin. This is a protein that stimulates the cells in the thymus gland, and this is a gland mostly active in children that helps to develop and establish the immune system. So this might be an area that would obviously have some impact on immunity and thus on asthma. So this was from the number 5 chromosome. The number 17 chromosome, there's a number of genes in this area that scientists are not sure yet which particular ones might be actually the culprit, if you will. But there is one of interest here that has to do with the role of calcium signaling. And calcium, of course, is in all of our bodies and is very important for many things, but cells also use it as a message to turn on certain functions of the cell, sometimes communicate with other cells, and so on. And there was some excitement a couple years ago, just 2 years ago, reported that there was some changes noted on a gene on chromosome 3 that did relate to calcium signaling like this in cells, and they were actually able to use that to develop an intervention, a treatment, which included a medication that has been used to treat osteoporosis, which naturally has to do with calcium metabolism as well. So I think this is an example of how our genetics may not come up with a cure for something, but it does lead, it gives clues that then lead us in another way that may develop information that's important in terms of treatment or prevention and so on. So we haven't tested this particular region of the number 3 chromosome yet, but this is the kind of thing that can be done in the future. I'd like to make this is kind of the end of the science part. We're still working on lots of data that we've collected and have much analysis left to do. But I'd like to skip over a little bit into some of the spin-off effects of research. We've talked a lot about it here today so far, but this is one example here. I think some of you have met Joe Ursheta. He works for Missouri Brakes, and a year ago now, we were able to obtain funding through the NIH Minority Supplement Mechanism through MHD, and so he was able to obtain funding to do additional analysis on the blood samples that were collected from these children. And so this is able to leverage Joe as a very experienced and knowledgeable individual, both in genetics and research in general. It leverages the instrument that was purchased for Missouri Brakes and is located in Eagle Butte, which will be used in this process and can be used by many other researchers as well. And it also involved Sanford, in that Sanford was kind enough to allow the samples that were collected to be maintained here in the sample repository for the exclusive use of our study. So lots of people involved, and congratulations, Joe, and thanks to NIMHD and CRCA for supporting this. I think it's important, as many people have said, that we translate the findings to others, and this goes out to many different individuals. We've presented our findings, some of our findings, to the local medical staff at Eagle Butte. The local community, through their health director and so on, have reviewed and approved our manuscripts so far. We've had presentations at the Association of American Indian Physicians, participation in a research conference at Eagle Butte. There's been 2 of them so far, another one next week. We've also engaged with students at InMed in Grand Forks. These are American Indian students in medical school, with high school and tribal colleges and so on. The students that have worked with us and have done the genetic analysis up at Turtle Mountain on these samples from the asthma study have presented their results at various conferences, and will be presenting actually on Friday up in Fort Totten as well. One of the somewhat unique programs that we did start with this, again thanks to Amy and others that had confidence in us, we purchased a small instrument that allows this genetic analysis to be done, and we basically created a small traveling roadshow, I call it, where we've gone out to various tribal colleges, to summer enrichment programs and so on. And if the students are interested and willing, they collect some of their own DNA from their saliva, They test their ability to taste a particular substance called PTC. This is a common test that's done in biology classes to demonstrate genetics. Some of us can't taste the substance. Most people, it tastes sort of like a bitter aspirin crushed in your mouth, but the ability to taste is determined by the genetics that you inherit. So the students are able to test their own DNA to see what their genotype is and how that compares and correlates with their ability to taste. So it's a very direct and I think powerful way to show the power of genetics, at least in a simple system such as this. We also discuss the ethics of doing genetic research. We talk a little bit about the potential career and business opportunities in genetics for the students, and I think it's been generally very well received. We've done this for 3 years now. Our biggest year was in 2015. We're in touch with 57 students that year. Last year I was traveling and couldn't do very much. We have 6 students contacted at NDSU at that time. But we have plans to expand that as well, and I'll go into that in just a minute or two. As I mentioned, we have published our findings on immune measures. The genetic effects I showed you are under review at a publication. We hope to have them accepted soon, and with a little bit of luck. We have a number of other studies or analyses underway in terms of indoor air quality, pulmonary function testing results that we have, the social determinants of health and how they relate to asthma, and the methods for recruitment and retention. So all of those things will be, we hope to have papers available shortly. The genetic analysis in particular depended heavily on the infrastructure that was begun as a part of a preeclampsia study that we developed at Turtle Mountain Community College starting in 2004. We developed the capacity to do the kinds of genetic analysis that what we call genotyping, which allowed us to make comparisons between women who've had problems with toxemia or preeclampsia and those that hadn't. We have currently a little over 550 women who have helped us with that study so far. But it really depended on the Turtle Mountain Community College students who were involved with obtaining informed consent after proper training and certification. They collected samples, they analyzed the DNA in the laboratory, they assisted with analysis of the results and preparation of manuscripts and so on, and helped to teach other students about their experience. Over the years, we've had 37 student research assistants so far involved with the program. 21 of them gone on to complete bachelor's degrees or greater higher degrees in social work, nursing, secondary science education, physical therapy. One student is working as a physician assistant now, and another student is at the second year in medical school. Our first laboratory technician, something many of you know, Melanie Nadu, has been here and talked about before, but she was our first laboratory technician in 2004, and she will be receiving her diploma, her PhD diploma here this next month at University of Minnesota. So we're very proud of Melanie's achievement there. Moving along, we have another student which has joined us, Caitlin Lawrence, and we received funding through the Strong Heart study for a minority supplement for her to basically expand the enrollment of our preeclampsia study so that we are able to enroll individuals at Eagle Butte. We have permission for that so far, but we also hope to enroll individuals at Pine Ridge after proper approval has been obtained. But Caitlin is a second-year medical lab technician student at the University of Mary in Bismarck. She previously worked with Marcia and her group as an intern and so on with MBRI, with Missouri Breaks. And we've also obtained additional instrumentation for another thermocycler, which will be located at Eagle Butte, and will allow Caitlin to do the same kind of a genetic analysis that's being done at Turtle Mountain now. So I think you can see this interconnecting web of different studies that we've used to try one thing to leapfrog into the next one thing to support the next one, and we've tried to build on this to the best we can, and I think that's necessary in a rural environment like this. We don't have a large surplus of resources. We really have to do the best we can with what we have. This is just to give you a little idea what the instruments look like. We're not talking about a large atom smasher. This is a small toaster-sized piece of equipment, and this it's called a thermocycler. It basically, those little holes in the top there allow a small test tube to be put in there. It carefully adjusts the temperature in cycles up and down, and as the reaction occurs inside that test tube, there are laser beams that come down from that top part and goes into the top of the test tube, and that monitors how the reaction is occurring and allows you to determine what the genetic makeup is of that particular sample. Again, this whole networking part, just to reemphasize, this is Turtle Mountain Community College in the upper left, a nice shot of the landscape around Turtle Mountain down below there. But the laboratory and student research assistants at Turtle Mountain, of course, have been vital to our efforts. University of North Dakota was funded through the INBRE program, and that's what allowed us to do our preeclampsia research. Stanford University, we have a representative here from there as well today, but they've been involved more recently in what we call a sales study, and recently allowed us to purchase some of the additional instruments that will assist at Eagle Butte in training additional students. The Strong Heart study, of course, through NHLBI was helpful in many different ways, but most recently in funding the minority supplement for Caitlin Lawrence. And NCAI has also been very helpful in promoting and helping to fund some of the red talks that are called that help to explain epidemiology in general and some of these research projects. I can't really acknowledge everyone, but these are some of the main actors, at least with the asthma study. Ray O'Leary, who I mentioned, and Marcia O'Leary, who has been directing Missouri Breaks for many years, Joe Urasheta, Kendra Enright, and Tara Lynn Halfred. The Cheyenne River Sioux Tribe has been very supportive and helpful over the years, as is the tribal health department there. Turtle Mountain Community College, we've mentioned, the Indian Health Service, particularly at Eagle Butte, the IHS hospital and staff, of course the CRCA grant and Sanford Research, NIMHD, and the North Dakota INBRE program. I have some contact information there, but I think that's as much as I can tell you today. We're probably about out of time. So, thank you.